Okay, I think we can start. Just show a little movie about what is biological computation. And this is a very old movie and very famous one, but if you haven't seen it, you should see it. This, I think, is called a macrophage, and it's chasing a bacterium. These things are red blood cells here, I think. And so it's chasing the bacterium. It doesn't like, you also bump into this one, but doesn't no interest in that one. It wants to get this one. And it's pushing away the other cells and chasing and chasing and chasing. This is not real time. This takes us probably several minutes to actually execute. But you can see that there's something highly non-trivial going on here. And no, this is a single cell. It doesn't have eyes. It has smell, but no eyes. It doesn't have muscle, although there's something similar. Certainly does not have a nervous system, but is able to act as a predator and get something eaten. So this is an example of, and of course people don't quite understand how this works. So it's an example of the kind of computation that we are after, or at least maybe it's not computation, maybe it's an analog control system, completely analog and maybe completely built-in and specific, but it is a control system of some kind. So, so given that, I want to give you an idea. So we don't understand that kind of algorithms, how they work, but I want to give you an idea of at least an example of a molecular algorithm, which is not completely trivial. So we've seen min and max, so let's do something a little bit more interesting that we can run with DNA. And this actually comes from computer science, comes from the field of sensor networks. And the idea is that, the original idea was that you have a bunch of sensors, like temperature sensors, and you throw them into the field, and some fall into the garden, some fall into sunshine, and they think it's hot, and some fall into shade and think it's cold. And they have radio, they can talk to each other in pairs, one by one, pair by pair. And their job is to kind of agree whether overall is hot or cold. So they have to find out who is the majority. Does the majority think it's hot or does the majority think it's cold? And then they have all to agree that it's either hot or cold. So they have consensus. This algorithm was proposed in 2007 in a conference in Distributed Computing. It was given a set of kind of automata that talk to each other by radio, so stochastic, it's randomly pairing and so on. But we can write it as in our chemical reaction language. It's quite simple. So imagine how they could try to coordinate. Suppose hot talks to hot. OK, we agree it's hot. Cold talks to cold. We agree it's cold. But then hot talks to cold, and what do? They could do nothing, but then the thing probably does not converge. Or they could decide to swap. Again, that doesn't do anything, probably doesn't converge. Or could randomly go to both hot and both cold. So none of this is very good. But there is a much better algorithm, which in fact turns out to be optimal, which is to use 3 states, hot, cold, and undecided. And I can write it as reactions. So if we say X is hot and Y is cold, for example, so hot talks to cold, and they don't know what's going on because they don't agree but do not know what the majority is, so they become undecided, both of them in this case. And then undecided talks to hot and says, oh, he's not cold, he's not undecided either, so he must know something, so I'll follow him, and becomes hot. And undecided talks to cold, says follow cold. So this is all. And this is optimal for this class of problems. It converges in logarithmic time, pretty amazingly. And we can write it as a chemical reaction network. So if you actually simulate it, this is what it does. It's interesting. So suppose we start. So in the best scenario, there will be some actual majority, and that the majority will probably win pretty quickly. But this also works, it's stochastic, also works when they are 50-50. So let's start in the worst case scenario of 50-50 here. And what happens is that hot and cold start talking to each other a lot, because there are many the same size. And basically they converge to a 1/3, 1/3, 1/3 situation of hot, cold, undecided. And then the system stays there apparently thinking for a while until randomly, because again it's stochastic, one of the 2 sides gets a little advantage, and then there's an exponential ramp up, which is due to essentially this autocatalytic reaction here that is amplifying itself. And then it very quickly gains the advantage, and everything goes in this case, let's say, hot, okay, and the other 2 disappear. And this is optimal. The conversion logarithmic time is with high probability. Also is robust, meaning that if there's a perturbation at this point, it does not switch randomly to the other side. For a big fairly large perturbation, it will not affect it of this size here. So it has a lot of quite interesting properties. And this algorithm has been found in biology in more than one place. There is in methylation and acetylation of DNA strands. They run this algorithm to decide uniformly to methylate or acetylate. And also it has been found in cell cycle, various cell cycle mechanisms. So it's interesting that an optimal algorithm shows up in nature quite a bit. And we actually implemented this in DNA. Well, this is again, the movie showing the actual helical DNA. You get, in this case, that reaction is going to run. So first X comes, displaces, then Y comes, displaces, and then go to the other half and displaces the 2B. So we've seen that before. So we actually did this as an experiment. So we designed the strands in the way I've described, the two-domain representation for these three gates that we had, these three reactions. And we mixed them up into some salty water. And this is for in the case where hot is 0.7 and cold is 0.3. Run the experiment. The solid is the experiment, and the dashed line is the predictions, I think. So close enough. And as I said, there are lots of uncertainty in the kind of quality of the DNA we get from manufacturers. So this is not too surprising that we're a little bit off. Now, in general, what I want to do in this section, in the first part, we talked about the promise and the general trends in this domain. And now I want to talk what we need to do to actually get there in the next 50 years, because this technology has a lot of potential to transform society and do lots of good things. And so we need some kind of plan to how to get there. And if I look at the state of the art in this domain, I compare it with the state of the art of computing like 50 years ago or so. So there are lots of analogies, and different parts of this enterprise are at different stages of tardiness. So the mathematical foundations, which I will probably talk about later, they're like concurrency theory in the 80s. So there are things we know, there are many things we don't know or understand how to do. The programming languages, we are basically using assembly language, chemical reactions here to engineer systems. We want to do better than that eventually. The analytical methods, they are getting pretty good. We can do stochastic model checking, all kinds of calculus. There are lots of good tools if you know how to use them. The worst part is the actual device architecture and manufacturing, which is like transistors in the 60s. They're flaky, they work after the time, they leak, they have all sorts of problems, unreliability. People believe that DNA goes bad after a week. Some people believe it goes bad after a month. So nobody really knows what's going on. And so again, we need some proper engineering of this kind of devices, which nobody has actually really done yet. But it's getting there because of the interest in DNA storage. Now people are really pushing, like Microsoft is really pushing the DNA producer to get really good quality DNA because they're paying good money and they really want good DNA for that kind of application. So there is something going on there. Also, we want to realize this potential of molecular programming in a kind of real engineering way, and this is what my friend David Solveig says, with no alien technology. So what is alien technology? It's proteins. So proteins for us, it's alien technology. We do not know how they work, pretty much. They do not know how to engineer them, how to predict them. What is different with DNA is that since 20, 50 years ago, there are very good algorithms. Given a DNA sequence, they will tell you how DNA folds. Okay, predict the secondary structure. That can be done pretty well, not perfectly, but pretty well algorithmically, so we can predict what we're going to get. And that is certainly not true to that extent for proteins. Even given the sequence of amino acids, folding problem is still quite difficult. So proteins, alien technology, we don't want to use them if we don't have to. And all the rest is basically a software problem. Now, chemistry, as I said, we would like to use that as a programming language. It is a concurrent language, or the reaction goes in parallel. So it has a connection with the theory of concurrence. In fact, chemistry and Petri nets are pretty much the same thing, and stochastic chemistry is pretty much the same thing as stochastic Petri nets. In fact, apparently Petri, Mr. Petri, got his idea of Petri nets from chemistry. Actually, he was trying to model chemical processes. So there is a quite interesting connection there. So, and there are lots of techniques we can use from theory of Petri nets for analyzing the stochastic version of these systems. And there are also process algebra, which is fancier version of Petri nets and so on. And we can make connections between these various areas, which I will not go into. But so there is a good chunk of theory has been done, and a good chunk of theory still needs to be done, and there are people working in this area. So this is a good direction to move forward theoretically. In terms of systems engineering, we want to have programming languages. So at the moment, chemical reaction networks, like for that, you could start with Petri net, almost exactly the same thing. Or you could have Boolean networks, that's a little bit different. Or you could have some other language that you may invent. And then at the bottom, you have architectures like Intel, Motorola, something, computer architecture. Here we have DNA architectures like 2-domain, which I've talked about, other representations of signals, 3-domain, 4-domain, and so on. So if you think of them a different computer or different device architecture that you want to target, and when you do that in a compiler, you try to invent an intermediate language to factor out the complexity of going from n places to n places. So we actually design these intermediate languages that take high-level languages and can map to multiple architectures. We'll probably describe one of them a little bit later. So this is almost classical compiler technology, assuming that you're going to have develop higher and higher level languages. These higher level languages, to some extent, they do exist. They've not been developed terribly because mostly they are applied to writing one paper. They're not applied to writing a complicated big system. But for example, one programming language is that of Boolean networks for genes, to model gene networks, and they work amazingly well with respect to experiments, surprisingly well. There are different kinds, synchronous and asynchronous, and they all work amazingly well. So for some mysterious reason, gene networks sometimes are very, very simple. Sometimes not, but sometimes they are. And for protein networks, we have theories of concurrent interaction, so like things in a soup bumping into each other. Again, we can use process algebra, and there are more specific concurrent systems which have been developed called graph rewriting, and they're actually used in practice for modeling biochemical systems. There's a system called BioNetGen, which is in quite wide use, which is isomorphic to one of the more theoretical systems. And in about membranes, there's a whole area of membrane computing, which talks about what you can do, what membranes can do when have some kind of active capability. And also I participate in these kind of communities to some degree. So there is a lot of potential development for higher level language, and this is my ultimate goal, my ultimate dream, to look at this as a program. This looks like a picture, but it's really a program. Explain the program that a virus has to replicate itself, to invade the cell and replicate. And this involves basically everything. It involves genes, proteins, and membranes. So just to give you an idea, this is the DNA or RNA of the virus. There is a protein capsid, which is the red thing. And around the protein capsid, there is a membrane, which is the same kind of membrane that the cell has. And there is some magical green protein sticking out on this virus membrane. Now, you would think that the virus could just merge with the cell, so it could enter the cell just by merging with the membrane. That's not what happens, maybe because the cell has defense mechanisms. So typically, the virus lets itself be eaten by the cell. And normally, the cell would then send it to a compartment where it gets disassembled. But these green proteins play a trick on the cell. They're able to perforate the protective cell membrane and therefore escape into the cell. So those are the trick of the green proteins. Once the capsid is inside the cytoplasm, it can break apart and the RNA escapes and starts replicating. It replicates copies of the protein capsid. It replicates copies of the green protein that it needs later, and it replicates copies of itself. And then all these three different things come together to reform the capsid inside the RNA, and they bud out of the membrane. So this green protein gets built in a special place that eventually merges with the cell membrane with the green proteins in exactly the right orientation, so that when the cell buds out, it reconfigures exactly in the original configuration. So all this is an algorithm. So it's hidden in the virus RNA. There is an algorithm to do all these steps, and this algorithm involves basically the whole cellular machinery of genes, proteins, and membranes. And so and this is a picture. It would be nice to have it kind of in formalized language, be able to express this kind of configuration and computations. And what I like, so this is typical of what you find in biology textbooks, by the way, and almost like one of those Renaissance paintings where you have the saint doing that, then the saint doing that, and the saint, you see the saint 7 times in the same painting. It's a history of it. It's a series of snapshots. So this is what happens, is the virus in 7 different phases of its life. So, and this is what we are trying to do in my lab in the long term in Microsoft in Cambridge. So we're trying to build a platform for programming biology, and we have various strands. The DNA programming, which are what I'm talking about today, but also a more classical synthetic biology strand, and also a strand about programming stem cells. And, but we see this as building a platform, so there's a lot of commonality in all these activities, and mostly resolve into mapping all these kind of high-level activities down to chemical reaction networks, and then doing something with the chemical reaction network, like simulation, analysis, and so on. So there's a lot of common infrastructure that we're trying to build, making sure it's compatible with all those things. And then we want to map this common infrastructure to maybe lab equipment or whatever you need to do experiments and so on. So I'll give you now an example of what our tools allow us to do for designing DNA algorithms, like the algorithm which I used to implement the transducer. So this is a tool, we call it DSD, Visual DSD. Visual comes from, it's not visual at all, it comes from the Visual Suite of Microsoft tools, but DSD means strand displacement, DNA strand displacement. Basically, we have a mysterious language, so this is a snapshot of the tool, focusing on one of the panes. We have a very mysterious language, which is this one here. Don't try to read it. All it does is, in some mysterious way, it describes these pictures, these structures. So this is just the output of parsing this strange notation. Basically, we have a way, syntactic way to describe in characters to describe these two-dimensional structures. And so we can describe the initial state of a DNA system this way by the various components. There's nothing that runs here. It's just describing the static structure of the strands. Okay? But then, once you have this static structure, by the rules of strand displacement, you can begin to figure out what interactions are possible. Not only the first ones, but also the ones that come after the first ones, and so on, and recursively. And so just from the structure and the rules of strand displacement, we can build a chemical reaction network of all the possible interactions from this particular initial configuration. And so this now gives a chemical reaction network where the species are the DNA configurations. Given that, this is just another representation of the same chemical reaction network as a graph of interactions between nodes. Also can export it to various other tools for different kind of processing. And then we can do simulation. The typical kind of debugging activities do simulation. Can be deterministic, stochastic, probabilistic, various kinds of approximations. But basically get an idea of what this dynamical system is going to do when you run it. Beyond simulation, we can try to do some kind of analysis. For example, again, if we start from and this you can only do for a very small number of molecules, so it's really for debugging, not for actually try to understand what might actually happen. But suppose you have one copy, just one molecule of each one of these 5 things that can interact in a very small way, but you can get the complete graph of all the intermediate states of this computation, and there can be some forking, and you can zoom in and see the transition probabilities given by the reaction rates of each of these transitions, and you can eyeball to see if there's anything strange going on here. For example, something that could be strange if you end up with the 2 terminal states, If it's only one terminal state, the computation is going, maybe it's forking, but then it's rejoining, there's some interleaving, and then it's converging. If you know with two terminal states, one of them could be a bug, something you didn't consider, and it's going off in a strange direction, and it stops there. So that's one way to debug, a simple way to debug some basic mistakes. And there are lots of mistakes you can do here. This is incredibly concurrent, incredibly combinatorial kind of computation. It's very, very easy to make mistakes, to do things that you do not think are possible, and they happen. And so you really want to debug these things as much as you can. Further than that, we can do, if you manage to build the state space, maybe now you use maybe 10 copies of each, it becomes a much, much bigger state space. You cannot eyeball it anymore. But you can feed it to a stochastic model checker, it becomes a continuous time Markov chain, probably a very big one. But you can feed it to a stochastic model checker, and we can compute probabilities of termination or probability of various events happening or not happening. So this is all standard technology which has been developed for other reasons in computer science, for applications to performance verification and so on. And that we can use here for this kind of system, because the underlying mathematical model of continuous time Markov chain is the same one that is used for these tools. Further in the future, we maybe can do some proper verification. So software verification is working quite well right now. We can actually formally verify with the progressing theorem provers some very complicated programs, even real production programs, C programs and so on. But in this area, everything is quantitative. There are transitions, there are probabilities, there are rates. It's not discrete like in software. So the verification problems become much, much harder. And you always end up having to do some verification up to some delta, some approximation. So this is still work in progress. But this, for example, is the work which I showed you before from Caltech, largely, was actually the system was developed using some of our tools. We share with them, and they share results and insights with us. So this system I showed before, they were analyzed before they did the experiments using our tools as well. So this is how you do design. So you have a computer support for designing and debugging, to do preventive debugging and so on. Then you actually want to execute this in a wet lab. So how do we do that? Well, so suppose we've done the design and we've debugged it and we think it's correct. And now we want to build this thing here. We want to build these five strands. So how do we get molecules out of this design? Well, the first thing you do, first of all, these are domains. So abstract domain X, A, T, Y, we do not know what the sequences are. We have no idea. So the first thing we do, we go to Caltech. They have a tool called NUPAC, which solves the reverse problem. They're given a structure, it finds, by kind of thermodynamic analysis, it finds sequences that will hybridize or fold into the structure that we designed. So you give it a structure. So this structure here is exactly the same, different representation of that structure there. These sticking out things are those sticking out things. And this tool is able to, by a search process, to find sequences, which are this one here, that will hybridize into that configuration, but remain orthogonal in other, so there's still orthogonal domains there. And it can give you multiple solutions, of course. There's not a unique solution, but you can ask for one or two or three or more and so on, and you see what you think you believe. And now we have sequences. So now we have some at least candidate sequences that we can use. So the next step, you go to a search engine and you type DNA synthesis, okay? And the first hit after advertising and Wikipedia is Integrated DNA Technologies. So these companies all over the world, and they make DNA. So if you order, so what you do, you go to your website and you take the string got from Caltech and you paste it in their form. You pay with credit cards if you have an account. And if you order by 2 p.m. today, anywhere in the world, by tomorrow morning, by FedEx, you get your DNA. There it is. And you get it in the mail. It doesn't say biohazard, because in fact, it's not biological. It's completely synthetic. It's not even wet. It's dry DNA. Each vial is a different DNA sequence. And it comes with a specification sheet. It's funny because it always says that DNA is perfect. It never says there is any defect, but we know that's not true. Anyway, so what do you do next? Well, how do you run those? Now we have the molecules. How do we run it? Well, the first thing you have to do is to add water. Remember, that's a very important step. So you add water, and first you mix this now wet DNA to make the gates. So you need to let it settle a little bit so the gates get hybridized properly. And then you mix the gates with your input. Now, when you do that step, you put the gates and the input very quickly. So you put the input very quickly, put into a fluorometer. And so what I haven't said, I mentioned to somebody before, when you order the DNA, you can order it with the fluorophores attached to the ends. That these are molecules that will fluoresce if the DNA double-strand splits, and will not fluoresce if the DNA double-strand stays together. So basically, during your computation, if you end up separating these fluorophores, you will get a light signal, fluorescent signal. And that's your output. You have one bit of output, okay? There's typically, maybe 2 colors if you're lucky, but typically you get 1 color. 1 bit of output, that's your print statement, your debug statement. And so you stick this into the fluorophore, and you go to lunch, because it's going to take a few hours. And then pretty much always something happens. So you will see something moving. Fluorescence will change, and you see various curves. Typically not what you expect, but something will move. It's not just dead. It's something really happening, which is the first interesting thing. And then you decide whether this is what you expected to happen or not. Typically it's not. And so what do you do? Well, one thing you can do is a core dump. Now, a core dump in this business is called a blot. So you take your DNA and you pass it through an electric field that will separate it by size. So on this side, you have, this is just a calibration scale. These are the known length of DNA strands that you have there. So you know this is 40, this is 60, 80 bases long. And this is your test tubes. And from this, you try to measure the length of the DNA you got in your core dump. And from the length of the DNA you got, to try to reverse engineer what actually happened. So this is not an easy process, and typically you have very little information. But you try until it works. Eventually, maybe it works, and then you publish, and then you're done. Okay. So this is the process. It's quite fun. You can do this in a week. I've done it in a week. So you can go on Monday and Tuesday. You design your system. On Wednesday you order it. On Thursday you get it in the mail. You go to the lab. Overnight at least. Certainly you can run it. And Friday morning you get the results. So and then you go back home and you try to figure out what really happened. Okay, so I want to talk about applications. First, some fun applications, and then some more serious applications. But first, look at the fun side. What can you do with this technology? So this is still hypothetical, but something which is very much in everybody's mind is to use DNA gates to inject them into cell, and either DNA or RNA gates, depending on what is more appropriate, and use them to do on-the-fly RNA rewiring. So, when the cell transcribes genes, they get transcribed to RNA, a messenger RNA, which means the message is going to other genes, and you can kind of intercept those messages and replace them with your own messages. That's what a gate would do. You would take an input, so therefore subtract the existing message and replace it with another message. So you can hotwire your cell and convince it to do something different what it was trying to be doing. So this is very appealing also because it's not going to change the genetic program of the cell. It's just external intervention without modifying the genetic program itself. So this is something which seems very general and might have a lot of application, but it's still quite hard to do in practice. This is something that actually works. So it's very difficult to get DNA running in live cells. The cells have, they do not like to have DNA injected into them because usually think it's a virus, and so they immediately try to kill it. So the cells have a lot of defense mechanisms against foreign DNA, foreign RNA. And so people are trying to get around that in various ways. They're putting DNA in protective cages and protective membranes, lots of possible solutions there. But for the moment, what really works is to do that in dead cells. So dead cells are not going to try to kill your DNA, but it's still extremely useful because remember the hybridization chain reaction? So you can arrange it so that if there is only one molecule of what you're trying to detect, you can cause this chain reaction to start triggered by this individual single molecule of target, and that will make something big, something visible on the microscope. And you can actually, you have fluorophores of different kinds attached to this DNA that gets polymerized, and so you get different colors showing up in your cell at the same time. And the fact that you can color with multiple colors in the same cell, that's a breakthrough technology that does not exist by and large. You can usually stain with one color, maybe two, but being able to do 5 colors is completely groundbreaking. Now you get correlation between 5 different, completely different quantities you're trying to understand. So these are examples of this kind of coloring done by DNA hybridization chain reaction in various kind of organisms, and this is the cut-tech technology, which is now quite well developed, and it's really a breakthrough technology. Live clothing. So this is surprising. So this is more in synthetic biology. They've engineered E. coli, I think, in this case, so they will react to humidity and temperature. And so when you get too hot, the vents on your vest will open up automatically, and you get cooler. So this is some unexpected application of synthetic biology, reprogramming. Now you have to go watch this video, Hacking Yogurt. This is Tur van Baalen is going to explain to you how to, so what is yogurt? Yogurt is a cell culture, okay? It's a cell culture you can grow in your fridge overnight, and you can actually go on eBay and buy a few pieces of simple equipment. The most expensive one is something that gives an electric shock to a cell so that you can inject DNA into it. And so you can reprogram, you can insert genes because you can order from IDT or whatever gene you want, pretty much, not all of them, and inject them in your cell. And so you can do a cell, I forget what, yogurt, you can make it color orange if you like, if you inject the carotene gene, for example, or you can add the gene for Prozac, and you get yogurt with Prozac in the morning. So whatever you want. And in general, there's this incredibly fun competition called the iGEM competition, which has been going on for maybe 10 years now, and now it has like 300 teams every year of grad students, undergrads, and everything in between. They're competing for, it's a hackathon, so it's a synthetic biology hackathon. So they're competing for projects, and then they go in a lab in a week. They do the project, and then there's a result, and they give them prizes. And these are some examples I like. So E. coli is one of the workhorses of biology, but it smells really bad. So a student said, well, why don't we make it smell better? So we inserted genes to make it smell like bananas instead of what it naturally smells like. Somebody did a detector to detect when a piece of fruit is fresh or not, so you can know when to eat it. And some people from Ghana, they figured out how to use bacteria to mine for gold, because they have gold in Ghana, but it's a very energy and polluting process. And so incredible amount of creativity and ideas just from undergrads and grad students. They should go to the website and look at the list of projects. It's amazing. Okay, so now more seriously, I want to talk about markets, so real money, but the connection between that and scientific discovery, and the connection between that and molecular computability theory, because I think those things are very, very closely connected, and I will try to rationalize why I think they're connected and why I tend to work, of course, more into this area here, but I think there is a quite direct connection. So first of all, the application, the potential and actual applications of synthetic biology and reprogramming biological order in general, it's absolutely amazing. So you probably know that Apple and Amazon are on the way of becoming trillion-dollar companies, but this is technology is just starting right now. It's already 1/3 of that. And in the US, you have this split almost evenly in three parts. These genetically modified crops, you know what they are. They are the biologics. So the biologics are the drugs that you produce by biological means. So you probably do not know, but now, today, the majority of drugs is no longer made by chemical means. All those plants have been shut down, and the majority of drugs is done by bacteria or yeast or whatever, by biological means already. So this is a complete transition in the industry. And the other things are various kind of industrial applications, like biochemical products of whatever kind, and biofuels, food and agriculture applications, whatever. So these are kind of most like chemicals you would maybe once produce by, again, chemical means, but now you produce by biological means for industrial applications. So this is going to be a huge market, and these are the startups. List of the startups as of maybe a couple of years ago. Some of them are never heard of, so maybe they've disappeared, but some of them are quite well known, like Ginkgo Bioworks and Twist Bioscience that do DNA synthesis. So there is an incredible activity in this area. And just to give you an idea of some ongoing success stories, so Juno Therapeutics, what they do, they did some trials, and they had 90% remission in terminally ill leukemia patients obtained by reprogramming the patient's own blood cells, and that was amazingly successful. So this is a quite dramatic breakthrough. Amiris, they make jet fuel, which is actually using commercial flights from I don't know what kind of bacteria, from yeast. I guess. And they also make anti-malarial drugs, so examples of Biologics company. Ginkgo Bioworks, this is the equivalent of the fab lines. So I said there is no Intel for DNA computing, but there's beginning to be an Intel for synthetic biology. So you go to them and you ask them, please build me this circuit, and they build the circuit in some bacteria, and they give it to you. So that's a fab line, generic fab line. I mean, they have their own constraints and their own, preferred ways of doing things, but the idea is that they want to build fab lines for synthetic biology. And Modern Meadow is a company that they want to eventually grow meat in the lab, and that's not quite there yet, but they're growing leather, and that's already a huge market, and you will never need to kill cows in India anymore, so just get leather from the lab. And so this is the kind of some of the most successful, most prominent companies. Now, so that's going to happen, but the problem is that there is some low-hanging fruit that these startups are capturing quite effectively. But there is a fundamental problem that to make this industry really take off, you need to develop this application very quickly, and you need to basically to scale up scientific discovery, because there are so many things that we do not know about biology that we cannot take advantage of. And the problem with that is that there are way too many proteins for anybody to study exhaustively. This is one of the interactome databases, and just between interaction between HIV and the human proteome, you have 1,500. So who knows what's going on there? And this one is, these are the bacteria in your gut. They have 9 million unique genes that are way more than the number of genes we have. And all of them interact in metabolic ways with our own proteins and so on. And who knows what's going on there for metabolism. So, and how do you discover what, so you see 10 to the question mark interactions, they have no idea what's going on. So how do you unravel this complicated mess? Well, you need to speed up science. You need to speed up scientific discovery. And it's difficult, but fortunately there's a new kind of virtuous circle that is developing, and that has to do with lab automation. So as we learn to program physical and biological matter better and better with various techniques, including DNA computing, this will really transform the process of scientific discovery as it's understood today. Let me try to explain that. So what is scientific discovery, scientific method? Well, in the old days, there was just one guy, Mr. Galileo, and he did everything. So he was making observations and thinking of interesting questions, making experiments, collecting data, developing general theories, and publishing. So just one guy, Renaissance, they were doing the whole scientific method. Today, it works through collaboration. So the unit of scientific method is no longer one person, but it's one lab. And that means that classically, so before systems biology, classically there would be one lab working for 30 years, maybe 30 people, to study one protein to death. Okay? And then they would know everything about that one protein. The problem is that even just humans have over people don't really know, over 100,000 proteins. So obviously this does not scale up. And systems biology was really addressing this kind of problem, that this approach does not scale up. And so now we have system biology basically introduced the idea of high throughput data collection, which now hooks up in the supercomputers discussion we had the first day. So there is high throughput data collection, lots of data. You try to do everything in parallel in massive scale, and you go through, again, the scientific method cycle at this kind of highly parallel massive dimension. But again, this does not work well because there are so many things you need to be doing and so few people that can do them. So you want to automate as much as possible this cycle. So what people are trying to do now is to, in the limit, no, this is not there yet by a long shot, but in the limit, you want to automate every single step of the scientific method, including inference, now, there is an inner loop to the scientific method, which is to refine your theories. So this you can pretty well do mechanically. There is no big problem. Maybe by machine learning or something like that, you can do this. Now, developing general theories is a little bit further out. So that but no, it's not unthinkable. For example, there have been some successes in this project called the Robot Scientist, which was a machine connected to an AI inference engine that was doing experiments and collecting the result and trying to figure out what the next experiment should be and to collect the best kind of information and go through the loop. And actually, they claim to have discovered new scientific knowledge. So it's not completely unthinkable that you can go a little bit also around the outer loop. But basically, you want to automate everything as much as possible. And eventually, what is the limit of this process? The limit of this process is that eventually the whole thing, the whole scientific method cycle will be driven by one program that jokingly here I describe as while true, predict, falsify. Okay? But everything is automated. And when everything is automated, it means that you're going through the computers up the top, and you're going through the organisms at the bottom, and they are in a feedback loop. Now, this has already been done in some places. And I need to explain why that's revolutionary, because this is the classical view of the scientific method and engineering method. So in engineering, even in computer science, you have a model or a specification, and from that model, you construct a system or a program or whatever, and then you try to verify that what you built is actually what you meant to build. And that's the verification process. So you go through this loop of construction and verification. In science, it's completely the opposite. You're given a system, a natural system, and you go through a process of discovery to build a model, and then you try as hard as you can to falsify the model to see whether the model matches the system the way you think it did. Now, in engineering, if your verification step fails, you're very unhappy. It means that you did something wrong, the system was faulty, and you need to rebuild it again. Okay? In science, if the falsification step fails, you're very happy because you made a new discovery, something you didn't know. Okay, so completely the opposite. So in this case, there's a fault in the system. In this case, there's an error in the model. So it's completely flipped. What is the truth? In engineering, the truth is in the specification, in the model, and the system is by definition always faulty. While in science, the truth is in the system, and this is classical Popper, and the model is by definition always wrong, and you try to refine it. So here you go through this cycle over and over and over again, trying until you pass verification, but you know that it's never ending because you know there are always bugs. It's just there are bugs you have not found yet, and so you just keep verifying, trying to find those bugs you have not found yet. And here, you go across this loop over and over and over again because you always hope to find something that you didn't know, something you can falsify and make a new discovery. So this is the classical view. Now, this is pretty much true in physics and classical engineering. What I find fascinating in computer science and biology that this does not quite apply to these two disciplines. And that's a situation that happens when you have that both the model and the systems, they're both so complicated that neither of them can be completely right. Now, when you build a software specification, that's often sometimes more complicated than the system you're trying to build. And the specification itself often contains what we call bugs, because we did not specify what we meant to specify. We specified something else. So when the complexity is such that it's not like a simple physical model, but it's a complicated beast, then both things can be faulty. And now the same in biology, the models that biologists build, they have thousands of components, and they have they don't even imagine that the models are correct, and they have no hope of ever knowing the full truth about the system either, because the system is so complicated. So it's interesting that biology and computer science are kind of sister disciplines in this sense. So the only thing you can do is to keep going around this cycle. And now, where is the truth? Well, the truth is in the middle now. The truth is in the connection between the model and the system. You keep determining to step, okay? And either of them can be wrong, and one of them is wrong, you try to fix the other, so you keep them in step. So this is the only thing you can do, and this is, now we are in Feynman territory. He famously said, actually wrote on the whiteboard, that what I cannot create, I do not understand. So he was a scientist, one of the best ones, but he was saying that it is not enough to do just discovery. You also need to build to make sure you understand what you have discovered. Okay, so this is, I want to connect this kind of maybe pessimistic view of the scientific method with the fact that in this new approach to lab automation, we need to go through this cycle of observation and experimentation with more and more complex models that they feed into each other into a cycle that goes through computers and biological entities. So the one program that you have that automates the whole thing is actually half running in the computer and lab equipment and half running in the organisms that you're studying, and you're continuously changing both of them. So because of this, I think we need to understand, if we want to have a good theory of these kind of systems, you need to understand the computer half, which we understand pretty well. So we know what it means to do algorithms for computers and what is the theory of computability for computers. But we also need to understand well enough the half that goes into the organism, the programs that go into the organism. And here we actually need a different theory of algorithms and different theory of computability, which is still being developed. And this I find quite interesting because all these results are quite recent. They're not the classic results of computability theory. And I think this will be progress here, and there's progress being made. And what we need to understand here is how biological systems can, might, and do compute. And this is something we pretty much do not know today. For example, if you ask, how does a signal propagate from the surface of the cell, where all the receptors are, to the nucleus, where the genes are, to respond to that signal? Nobody knows what is the encoding, pretty much, of those signals. Or you can ask, what is the encoding of the signals in the neural network? Nobody really knows how signals are encoded. There are theories, conjectures, but you will not find definite answer. So what we can do, though, we can make conjectures. So, for example, there is a part of electronics that's called stochastic logic. Instead of having Boolean gates, they have gates that work on probabilities on frequency of firing of spikes of signals. And you can use this gate to do like multiplications of two different trains of spikes. And then you can ask, okay, this looks like, this is my encoding of signals. There are trains of spikes. These are my encoding, these are my gates that do some operations that I can understand. And then you can go in the biological system and say, does it look like this looks like the gates that they have there or not? So you can make this kind of connection. So you need to simplify a bit to try to get a handle on these problems. And one of the simplifying steps you can do is to say, okay, biology looks like just a bunch of chemical reactions. We can assume that to some degree, more or less true. So the question now can become, well, what can you compute with just chemical reactions? Can you do a lot? Can you do a little? And then it's a simplified problem. It's not the whole problem. But if you understand this much, then maybe you can get that to extrapolate to the next step of complexity. So just asking this question, I think, is quite interesting to understand what to compute biologically, to understand how biological systems work, and eventually to understand how to automate all these systems and go to industrial development. So the last part, I will dig a little bit deeper into this theory of computation for chemical reaction networks. And unfortunately, there are two schools of thought. I don't know what to call them. But the first one is the following. So there are two interpretations of chemical reaction network, and I'll explain at the end how they differ. The first one is it's called the discrete state semantics. And here is where you believe in the atomic theory of matter, that atoms are discrete entities. They're not continuous things. So there is a number of atoms that you can count. And reactions are reactions where atoms collide, molecules collide, one by one, and they produce more molecules on the other side. So everything is countable, it's discrete. So that's why it's a discrete state semantics. So in this model, a state of the system is a finite set of molecules, okay, of different species. So you have 200 of this, 300 of that, 100 of those, but they're all finite. And you also have a finite set of reactions that operate on these molecules. So it's a multi-set rewriting system, basically. It's Petri net also. But the interesting aspect is that reactions have a continuous rate parameter. The speed at which the reaction operates is a real number and tells you, if you have two reactions competing with each other, it tells you the probability of one winning over the other at any given point in time. So the mathematical model becomes a continuous time Markov chain, where the state space is discrete, but the time moves continuously because you have these continuous rates. Now, the problems you want to have analyzing this kind of model, so you want to compute the kinetics of this model, meaning compute the distribution of outcomes over time. You want to analyze mean variance, other moments of the distributions, and you want to ask about state reachability. Can this molecule be produced? What is the probability of producing this molecule after a certain time? And so on. So these are the questions that you want to ask. And again, let's look at some examples. We've seen some of those in this discrete state semantics. These are some we've seen, and some even simpler ones, like multiply by 2, you just make 2 copies of x into y, and so on. So we've seen min and max, and it's the approximate majority. And the question is, you can write these programs, but how complicated can these programs get? What can you compute with this kind of model? And actually, the answer is known. It's a fairly recent result, but it is known that in this model of discrete molecules and finite set of reactions, you can only compute the semilinear functions. That means if you have a function like this of 2 parameters, that these are your species, and you want to do this kind of computation, you look at the graph of the function. If it looks like a union of linear sub-pieces, so this is a linear piece, that's a linear piece. If a union of linear pieces, it's called a semilinear graph. And all the semilinear graphs can be computed by chemical reactions, and only the semilinear graphs can be computed by chemical reaction. So for example, if you have a function which is squaring something that's nonlinear, cannot be computed. So this seems, one way, it seems quite limiting. The chemical reactions are, in a sense, so weak in this stochastic discrete model. But in fact, they're also surprisingly powerful in another sense. Suppose you did not know that result, that you did not know that chemical reactions were so weak, and you say, okay, let's simulate a Turing machine. Okay, let's do something interesting. Instead of a Turing machine, let's take a register machine, which is easier. So register machine has registers, a bunch of registers are 1 or 2 or 3. It contains a number, and the number of molecules in each species represent the number stored in that register. And it has 4 instructions. You have to be able to increment a register and jump to a new instruction, so I is the address of the instruction. You will be able to decrement a register and jump to another instruction. You have to test whether the register is positive and then increment and jump. And then you have to test for 0 and do something. So let's try naively to implement this Turing-complete device with chemical reactions we now know are not Turing-complete. Let's see what happens. So I can represent a program counter, so the address of a reaction, by a single molecule of species PCI. So if I have a single molecule of PCI, it means I'm at address I, and I'm going to execute that reaction. So now to encode this reaction, so suppose I am at PCI, so this reaction can fire, and when this reaction fires with a single molecule, it will add 1 molecule to R1, so increment in R1, and then it will produce PCJ, which means jump to reaction J. Okay? You have 1 molecule of PCJ now. Decrement. Well, now we put the register on the left-hand side, we need to have the program counter here, and we need to have at least one molecule in R1. And then we make this step that will remove that one molecule from R1, so the decrement, and also delete that program counter, but create a new program counter to jump to J. So that's all fine. This one is fine too. We can, if you're program counter I, we test that there is at least one molecule in R2, but we not decrement R2 because we leave it. But we increment R1 and we jump to PCJ. So all this is fine. But then we come to this one, test for 0. So how do we test for 0 molecules? Well, a reaction will only fire if there is something there to fire on. So any suggestions? Test for 0 molecules. So basically, this is where the Turing completeness fails. But these reactions have rates. So it turns out that you can have a notion of waiting long enough because you can adjust the rate of this reaction. So you can say, OK, I'm going to wait for 2 hours. If I don't see any molecule, I'm going to say, OK, there are no molecules in the register, and so on going on. And so you can build an approximation that is not Turing completeness, but can get as close as you want to Turing completeness, up to epsilon. So this is really a new class of computational devices, which I don't think existed before this result. And it's quite tricky because you need to fix the error that you want to achieve ahead of time, and then you need to run your Turing complete computation, and you need to stay always below that error, but you do not know how long you will run because that's a Turing complete device, so you have to basically slow down quickly enough to always stay below the potential error that you fixed at the beginning. So it's a bit tricky, but it can be done. Anyway, what this says is that although it looked like an incredibly weak computing device, this chemical reaction network, actually in practice you can achieve Turing completeness if you're willing to wait long enough. So this is the result about discrete semantics. Then the continuous state semantics is maybe what you learn in high school, that each chemical has a concentration, which is a continuous quantity, it's no longer discrete. It's a real number, and you still have real numbers for rates. And now here you can write completely different programs that you could not write in the other model. So here the rule of the game is you have the law of mass action that describes how the system evolved. It says that the speed of a reaction depends on the rate and also the product of the concentrations of the reagents. It's a multiplicative law. And here the issues, again, that you want to analyze computing kinetics, outcomes over time. This is typically deterministic outcomes given by differential equations. Analyzing equilibria or steady states and doing model reduction if you can do it. And here I will not, there's a whole set of interesting examples. If you're interested to look at this very nice paper called Sniffers, Buzzers, Toggles, and Blinkers, it tells you how to code up a whole bunch of nice, interesting little devices with very few chemical reactions. So the buzzers are kind of sigmoidal response. Something goes bzz when I say something. The sniffers are things that adapt to the different levels of input proportionally. Switches are something that have positive feedback in them. And oscillators, blinkers are something that have negative feedback, so you can build oscillators and switches and so on. So a whole bunch of devices that you can build into this model. And a simple example, a simple programming exercise is what I could give you is give me a chemical reaction network that gives you this nice symmetrical shape as output. And this is tricky because in chemical reactions, everything decays exponentially. If you have a unimolecular reaction, it's an exponential decay, so it's not symmetrical. If you have a bimolecular reaction, it's a quadratic law, it's not symmetrical. So how do you build, so programming exercise, how do you build an output like that? But the answer is simple, 2 reactions, and you can do that. You can try that. And you can check that the result is correct by computing differential equations and noting the symmetry there. So you can make oscillators. It's very, very nice little programs with 3 reactions. There's a lot of interesting oscillators. It's also interesting in not making oscillators because for synthetic biology application, clocks are very, very expensive things to have because they use a lot of energy, use a lot of material. They have to run all the time. They have to be distributed everywhere. So in electronics, you spend a lot of energy on clocks, and everything is clocked very finely. In biological organisms, you do have clock. You have circadian rhythms. You have cell cycle rhythms, but they're very slow. They only happen once every while. And all the rest is synchronized without clocks by local interactions. So fortunately, there is a discipline in computing called asynchronous computing, where, like in your cell phone, you will have some asynchronous logic trying to save power. And that's based on the notion that you do not want to have clocks everywhere, only where they're really necessary. And otherwise, you use a local handshake synchronization between individual components, maybe individual gates or some higher level components. And the key ingredient to achieve this local, cheap, energy-efficient synchronization is the so-called Mueller C element, which is a Boolean gate, but not, if you've never seen this before, it's a bit strange. It's a Boolean gate actually has one bit of memory inside, so it's not a pure combinational logic. And the way it works is if X and Y are true, then Z is true. If X and Y are false, then Z is false. If X and Y are true and then Y goes false, Z remembers the last time that X and Y were equal. So it has memory of the last time the output is equal to the last output at which both X and Y are equal. So it has a bit of memory, and you can see how this can be used for coordination, because basically this switch is only when both of them are switching. So it's waiting for both of them to change state before this changes state, and this is how you achieve kind of coordination. So this, for example, we showed you can implement with a variant of the approximate majority network. You may recognize this diagram here. It just has more inputs. It's a dual rail logic with high and low inputs. And this you can do to implement. There's memory because it's a bistable system, so the bit of memory is in the bistability. And this is just a fancier version where you use an additional rectifier to make sure the output is really stable and good. So this is another interesting little program for that. And other things you can do steady state. Remember how I said you cannot do square of a number? Well, here you can do multiplication in the continuous model, no problem. This is a very nice paper, Computing Algebraic Functional Biochemical Reaction Networks, that tells you how to do all these operations: multiplication, division, square root, addition. So for example, if you want to multiply, if you want to multiply, you use the two inputs to catalyze the production of the output. So the output is X produced, but the inputs are not consumed. And also you need to, this would cause X to grow forever, and so you have a degradation reaction to balance X down. And so by the combination of these two, the growth and the degradation, this stabilizes to a point where X, it turns out to be the product of the quantities of A and B. And if you put B on the other side, you get division. Okay, and if you do this, you get, this is quite complicated. I don't think I can explain it. You really need to look at the differential equations to figure out what's going on here. But this is compute the square root of the input A by using twice the output. So you can do all the arithmetic operations this way. In fact, and then you can compose them. And the fun thing in this paper is that you can compute the root of a polynomial by composing all those operations. You can compute b plus square root of b squared minus 4ac over 2a, the positive solution, the negative solution over there, flowing from the same inputs over there. So that's a lot of fun. So you can compute algebraic functions in this continuous model, which seems quite powerful. Further, it turns out you can solve all algebraic equations. So here's an example, algebraic equation. You want to find the phi such that 1 over phi is equal to phi minus 1. And that turns out to be the Golden relation conjugate. Okay, and this is there is a systematic way for any algebraic equation to find the corresponding chemical reaction network. This was my own solution, which I found independently. I think this is probably not the same that comes from that result. But these three reactions, which are represented by this network here, so there's a feedback, there is a back and forth, and there is a feedback. Will cause W to stabilize to this value phi, which is, so this is a network that computes the Golden ratio, basically. And you can see this very easily from the differential equation. If you compute differential equations and you set the derivative to 0 because you're looking at steady state, you end up directly with this equation that tells you that Y is that. Okay, that's not it. But you have to guess what the chemical reaction should be for that to work. This is another quite interesting very interesting recent paper from Kamash. And this is how to do integral feedback controllers chemically. So these are widely used in engineering for control systems. And they're typically digital, or look at the input and output, try to balance it, keep the system at a set point. And there is actually a very simple way to do them with a couple of chemical reactions. Not only it works simply, but also you do not need to know what your network is. This is the amazing part. So this is an unknown biological network, but you want the output of this network to be kept at a set point. You can put a little bit of controller in front of it that it will keep at that point with some mild assumptions, no matter what the biology is doing inside. So this is quite an amazing result, basically independent of your knowledge of the biology. So that maybe we can do something more in that direction. And finally, I want to point out that one of my own papers is coming out, and the idea here is to again do some kind of computation in the continuous model in this case, by taking a known library of components. So I can go to any textbooks in electronics and take any linear circuit for the moment, so resistors, capacitors, inductors. And they talk about voltages and currents, which are things which are both positive and negative, so you cannot map them directly to chemical concentrations. But we have a way to translate uniformly, algorithmically, any such linear network, this is a high-pass filter, to a chemical reaction network, which has exactly the same dynamical behavior up to an approximation, because it cannot be exact. But you can make it as exact as you want. So this is another way to understand what you can do with chemical reaction networks, because if you see this thing in nature, you may have no idea what it's doing, but if you get it as a translation from a high-pass filter and then you see it in nature, you may say, oh, maybe that's a high-pass filter. So that's kind of the idea. And finally, I want to finish this programmability part with some bad programs. So not all chemical programs are good. So first of all, something I have not mentioned, but this first program here, any physicist will object to. It violates thermodynamics, because any reaction should have a reverse reaction. Okay? So this is under the assumption that the reverse reaction is small enough that we don't care, but you have to basically say that. This reaction here, you can write it, it's a program, but this violates conservation of mass. And again, a chemist will object very strongly to this program. But it's fairly well understood that what this means really is that there is an additional reservoir of mass that you're not expressing, and that, so this would be A plus X goes X plus X. There is some big reservoir of A that gets converted to X, so mass is overall conserved. And also it's quite common in biology to not consider ATP consumption in this reaction, so you do not actually write the reaction for ATP, although you should because they're using energy. So these kind of abbreviations are understandable. And so if you look at the face of it, if you just compute the ODEs of this program, this will grow X exponentially and it will go to infinity, but in infinite time. So all this kind of can rationalize, but this program here, this is really bad. This is Zeno's paradox. Okay? This program will produce infinite outcome in finite time. So at time one, before time one, it reaches infinity. So this one you cannot ever hope to achieve. Violate finite density in the sense that the density of your chemical will become infinite, which is in this mathematical continuous model is not in principle impossible, but in the discrete model you can never achieve infinite density because you have finite size molecules, so you cannot pack them too tightly. So unfortunately, you cannot rule out these bad programs because I'm pretty sure it's undecidable to detect these kind of situations. So you have to basically accept them in some form and analyze it later. So I've talked about two semantics of chemical reaction, the discrete one and the continuous one. Well, are they the same? Do they tell you, as already hinted, that they're not? But let's see, of course they are related because the concentration and the number of molecules are related by Avogadro's number, so you always know how to translate from one to the other. But as mathematical models, they're not equivalent. Let me show you in a concrete example a situation in which they're very different. So these are, again, approximate majority, and I showed you, I think, this version of it, where if hot and cold meet, they both become undecided. Actually, the original version of approximate majority was this one here. It was a stochastic algorithm. So if hot meets cold, then randomly one of the two become undecided, but not the other. So there are two reactions, one for when X becomes undecided, and the other one when Y becomes undecided. And of course, X plus Y, Y plus X is the same thing, it's just right in different order. So now if you look at the differential equations of these two systems, they give you exactly the same differential equations. So from the continuous model point of view, these programs, they mean exactly the same thing. In fact, in our experiment, we implemented that, which is really the wrong one, in a sense. But if you look at the stochastic finite model, they are different because consider the initial state where we have one molecule of X and one molecule of Y. Well, then this reaction can fire and produces two Bs, and then it stops because nothing else can happen, nothing else can fire. While in this reaction system, if I have the same initial conditions, then either this one or that one will fire. And so either I go XB or YB, and then this or that one can fire. And in both cases, I go to either XX or YY. And so now here I have two possible final states, but neither of them is that final state. So they're absolutely different behaviors. So this is, so who is right? Well, this is, in a sense, well understood. This has been going on for a long time, this distinction between discrete and continuous models. People in the literature tend to talk about when it is adequate to use the continuous model. So first of all, pretty much the correct one is the stochastic model, the discrete model. But there are lots of advantages often to consider the continuous model because there are very good analytical techniques from calculus. So people talk about when it's correct or adequate to use one or the other. But what is, and that has been going in mathematics for a while, but what's interesting in biology is that biology has realized, actually quite recently, they used to rely more on the continuous model. They realized quite recently that the discrete stochastic model is very important because now we have devices that can inspect single molecules in single cells and see the stochastic fluctuations in the single cells, and those cannot really be explained with the continuous model, and you really have to take this discrete model quite seriously. Okay, so that was the current state of the theory of reaction networks. As I said, we have tools that try to operate in both modes, continuous and discrete, and we try to basically cover the whole spectrum of possibilities. And I want to conclude with some remarks going back to DNA computing and so on. And I'm not a historian like the speaker yesterday, but I will do a very, very short history of DNA. There was this discussion, when did computing start? So I will take a little bit further back. DNA was invented 3.8 billion years ago. And as far as we know, that only happened once. When DNA came on the scene, well, there was RNA before it, but when DNA came on the scene, it basically locked the market and killed all the competition and got the monopoly on Turing machines forever, okay, for 3.8 billion years. So nothing interesting happened after that, except maybe 2 billion years ago when we went from prokaryotes to eukaryotes, the cells with nucleus. Now those genes work a little bit differently than the prokaryote genes, but it's still the same DNA, it's still the same genetic code, nothing really changed there. So the next interesting thing that happened after that was in 1936 with the invention of Turing machines, because this is the first time in 3.8 billion years that a Turing machine discovered a Turing machine. Actually, quite different one, okay? So that was a significant step forward. And from that, we got transistors and all the history that we've seen the other day. But what's interesting to me is not the hardware. So the whole point of Turing machines is that the hardware does not matter. Any computer can simulate any other computer. Any program cannot know what computer is running on. A game that runs on, it cannot know if it's running on Commodore 64 or an iPhone emulated in Commodore 64. It's still the same game. So the hardware is pretty much irrelevant. What's important is the computer programming. The notion that now we can systematically manipulate information in any way we want. Irrespectively of the hardware, irrespective of many things, we can systematically manipulate information, and that is what has really changed society in so many ways. Now, on the other side, so this is the history of digital computers. On the other side, people got inspiration from digital computers to try to build biological computers, and structural DNA nanotech was invented in 1982, and the first DNA algorithm was run in 1944. It was this kind of NP-complete problem. And people said, great, now we're going to have DNA computers. But again, the hardware does not matter. If it's DNA or something else, what really matters is the molecular programming, the fact that we now can systematically or will be able to manipulate matter in any way we want. And so this is what is going to be the breakthrough of the next century, next century, the ability to manipulate matter in any way and configuration we want. And this is what's really going to change the whole game. So with that, some acknowledgements. Microsoft Research, Andrew Phillips is my long-term collaborator there. We have a little group, not a big group, but a nice little group. We collaborate a lot with Caltech, the Winfrey Lab, and University of Washington, the Seelig Lab. Georg Seelig also came from Caltech originally. These are the people in Cambridge, and some various collaborators that I just mentioned. And some resources you may want to access, including the molecular programming project at Caltech that basically bootstrapped a lot of this technology over the past 10 years or so. And Georg Seelig DNA Nanotech Lab, University of Washington, that is continually and doing lots of interesting stuff there. And most of all, there is a series of conferences, the DNA Computing Molecular Programming conferences, that contain all the literature you ever want to know about all this field. So if you want to know more about this, look at those proceedings. All the papers are there. They're really wonderful conferences. They're completely interdisciplinary. It's a really unique community. There are experimentalists that do DNA experiments, and there are lots of theoreticians doing theory about DNA and other systems, and they all talk to each other. It's quite unique community. So, and I think that's probably the end. And if you have any more questions, I'll take them. Thank you.
Thanks for the wonderful talk. Two very quick questions. So the first of all is, so when we are talking about in silico computations, you usually have sequence of steps. In here, if you would have, for example, like 2 millimolars of X and you want to add to 2 millimolars of Y, you would probably get 3.7 millimolars of something. So which strategies can we use?
So there is no sequential. What you need to do is to have a nonlinear transitions between steps. So what the genes would work by sudden nonlinear jumps to sequencing of gene activations. So you need nonlinearity to implement sequentiality, essentially.
Okay, and the other quick question is, how could we measure time with chemical computation? I mean, time in the sense that in computing it's very important to keep track of time if you want to do control systems, for example. You mentioned that we can harness the power of the rates to develop, for example, oscillators. But how could we measure time itself as a variable?
Well, so that's actually, biologics is a very interesting question because in development, especially in the early stage of development, when the organisms begin to make their body plans, that is an extremely reliable and predictable and deterministic process, even though all the components are stochastic and discrete and so on. So there are certainly some error correction, noise attenuation, all kinds of processes there to ensure that time is kept precisely when that really matters. So we probably need to learn a lot of that, but we know it's not impossible because it's done. And there is a group of people at Caltech that studied the specific gene development network, which is pretty big, for like, again, 30 years, and with 300 postdocs and postgrads. And at the end, they had this gigantic network of early development of sea urchin, and with all complete molecular detail of everything, but they were able to simulate it with a Boolean network with a fixed time step of 3 hours every activation. So completely deterministic.
Yeah, thank you very much for questions. Okay, it works. Thank you very much for the talk. I would like to refer to the part when you mentioned about the discrete model of chemical chemical reaction. I got the impression that there are some limitations of the functions you can calculate with the discrete model. It's not true. Because actually the continuous model is, well, a kind of simplification of the discrete model when you reach thermodynamic limit, right? So actually the discrete model, when your number of molecules is sufficiently large, should give the same results.
Yes.
So actually what you say works with extremely small numbers of molecules, where no physicist can think of using continuous model.
Absolutely, the thermodynamic limit will give you that, exactly. The fact is that in cells, like you have one copy of each gene, so that's one copy. So in that case, you need to consider the stochastic behavior. And many other things are, like there maybe you have 10 copies of some messenger RNA at any given time. You may have one proton, so you have on average, I've heard, one proton in each bacterial cell at any given time. So there can be quite small numbers.
We did a paper a year ago about classification with oscillatory allosteric reaction. And we found that, well, the predictions of continuous model work when you have about 100,000 proteins, allosteric proteins. If you go down, then
yes, of course. And so we use a lot linear noise approximations and these kind of techniques to try to estimate the level of noise in these systems. And so we try to basically cover the spectrum between continuous and discrete that way. Yeah. Definitely, it's a hot topic, especially because of these new techniques to analyze systems with very small number of molecules in cells.
Hi, thank you for your talk. I really like this parallel with the computing and DNA, and I like your sentence where the hardware doesn't matter. And in computing, for that to happen, you need operative systems and drivers to abstract the I/O and probably compilers. Can you make a parallel between this paradigm of abstracting the hardware in computing with the DNA area where you work?
Well, so as I said, there are multiple architectures you can imagine, and people have designed to implement chemical reactions with DNA. So I've shown you one, which is this two-domain, but you can have different coding of signals. So in this case, the signal was a one-to-hold and one domain. There's another where it is a one-to-hold and two domains, which have different characteristics on the to-hold in the middle. So you can have, given your architecture for the signal, that has implications for how the gates should work to manipulate those signals. And so that becomes a completely different architecture. So that's an example of a different hardware that does the same job of implementing the same chemical reaction network. If that answers, I don't know.
Can imagine the next step. Probably this is a good theme for a team, the hacker. What will be the operating system for that world, the compiler, the driver?
Yeah, so that's what Ginkgo NanoWorks is trying to build. It's trying to build an operating system which is a kind of an empty shell of a cell in which you plug your application programs and then the whole thing runs. They're actually trying to build this kind of cellular operating system.
Thanks a lot. I'm here.
Okay, sorry.
I hid in the front.
Well, the noise is non-directional.
Thanks a lot. I really enjoyed the, let's call it, alternative model of science that you propose, like on the third part of the presentation. Some people would argue that was always the actual model, that science and engineering are always combined in that way since the beginning of mankind, not even since the beginning of this.
Well, to some degree, but there are different degrees of.
Now, the issues that you emphasized about the complexity of biology and the fact that no single person or no single team can grasp all of it are close to similar issues we have with the big data and the amount of data that we're trying to process today. And so I've seen what you showed looked really similar to the approach that the Google made with AlphaGo, these self-training AIs and so on. My question is about should we look for a different? So should we try to apply that idea or that schema of the scientific process, scientific and engineering process, to our daily life? What I'm trying to say is, if you tell that you are not fully sure about your model or you're not fully sure about the system in a courtroom, for instance. So for instance, you're taking a DNA sample from a criminal and you're saying, yeah, my model is not, I kind of think this is the way it is, but we are inside of a cycle.
The course understand very well the probability of DNA matching, so I don't think that's an issue. If you can quantify the probability, then you're fine.
But this is it's broader than that, because eventually it seems that we're going towards a world of black boxes, where our approach to science or to knowledge should be fundamentally different to what we have today. So this idea that we will not even be able to grasp the entire model at the end, is it something that you foresee?
This is, I think, specific to certain disciplines. I don't think that physicists, for example, should adopt this point of view. They can live quite happily in the old-style scientific process. This is specific to disciplines where the models get so complicated that they no longer serve the function of abstract mathematical entities that they were supposed to be, and they become something else. And then we don't know what to do, and so we have to deal with that. And so that seems to happen in software a lot, because it's quite common that your specification is out of sync with your implementation, and it also seems to happen in biology a lot. And
how do you take political or ethical decisions within this frame?
I don't know.
Thanks a lot.
Yes, Leon from Delft University. Thank you very much for your presentation. I have a question related to the computing model. I come from the field of quantum computing, and although quantum computing is like Turing complete, etc. in theory, we actually foresee that part of the computations are still done, like as how we call it in a classic computer, and only parts are offloaded in some kind of synergy between the two. Do you see like a similar model in your case, where kind of small parts are offloaded, but there's kind of a synergy with a classic Turing machine type of machine?
Yeah, that's I think partially what I was saying about this mixed cycle. So because you can imagine, I think there are some recent articles, you can imagine that you have biological detector, maybe in your skin or something, that sends a signal, like a light signal, to an electronic device that something has been detected, and then the electronics does some processing and send a signal back to the biological device, and so you have a loop where some of the intelligence is in the more appropriate place, like in digitally, and other is in biology. So definitely those kind of mixed system, hybrid system situations, in a sense, they mirror the notion of cyber physical computing that is more on the physical side, where you have mixed computation and physical actuation tightly coupled. And here you have mixed computation and biological actuation, if you want.
Thank you very much.
I have one last question. Anybody else wanting to do a question? So these mechanisms open a lot of possibilities for ourselves as computing devices. So, for example, humans have really hard problems with detecting some chemicals in the environment which can be super harmful. Carbon monoxide, for example. We wouldn't detect carbon monoxide for ourselves, right? So do you see this as also as a way of us building extra capabilities on ourselves that totally fit, like plugins on our own bodies that totally fit our bodies, so they're not exotic, but they still give us these capabilities?
I'm sure somebody will do that. I mean, that will, you know, I don't know if it will be widespread, but somebody will try for sure. And it's hard to predict what kind of forms that will take. That depends on, you know, technology and innovation. But certainly be able to have, one of the goals of this, many of these projects is to be able to have a very simple environmental detector sensors that you can carry with you, maybe little strips of paper or something very simple that you can use to detect conditions very quickly in the environment. So now whether you ingest them or you put under your skin, I don't know, that's a different big question. But the notion of making highly portable detectors, that's definitely one of the main goals.
Okay, so thank you so much. Let's thank our speaker again.